Hello. Hi and welcome to Chapter 6, Part 1B on DNA replication. Now, I found out there's something very fascinating about the current app I'm using. I can change my boring background. I'm clearly trying to entertain myself because you guys are not here to bug me. Anyways, hi. Today is about DNA replication. You can see this DNA molecule behind me, sort of replicating, and you can see these two strands are separated, and it's all like funky looking and really cool and really sci-fi. And I am a huge nerd. I'll stop talking now. Anyways, hi. Let's go back. Let's start at the beginning of this wonderful process, shall we? Whoop. Okay, so step by step. Now, DNA replication is semi-conservative. So in full, we say it is semi-conservative DNA replication. And this particular process, as we already know, occurs in the S phase of interphase. We know this from last chapter, didn't we? Last chapter when we said that during S phase, one chromatid becomes two sister chromatids. Okay, and what's happening is actually one DNA molecule becoming two DNA molecule. That's why the number of chromatids always equal the number of DNA molecules in that particular cell. This process requires ATP. And before we start our process, let's gather our resources. Let's see what enzymes we need. There are three enzymes we need. Number one is helicase, and helicase functions to break the H bonds, break the hydrogen bonds to separate the two DNA strands, sort of like what is occurring behind me here. The two strands are separating in this fashion, and that is by an enzyme helicase. Number two is DNA polymerase. DNA polymerase synthesizes a new strand of DNA. So DNA polymerase is really what is building that new strand and joining things together by catalyzing the formation of phosphodiester bond. It also proofreads DNA. Proofreads DNA means to look through the DNA sequence and see if there's anything wrong with it, and to repair that before it's too late. Excuse me. Number three is DNA ligase. DNA ligase joins DNA fragments together, and sometimes you have structures like this, fragments between different parts of the strand. And we'll see why fragments form later on, and these also catalyze the formation of phosphodiester bonds. Polymerase and ligase do catalyze the same bonds, but they are different. We will see how and why step by step. Okay, so enzymes needed again is helicase, DNA polymerase, and DNA ligase. Okay, don't freak out. They all end with ASE. They are all enzymes. Anything you know about enzymes apply to these three things. So they all have active sites. They all have active sites. They are all globular and soluble. Everything you know about enzymes applies here. Okay, but don't worry about it. Let's go through the process now, step by step. Now, the first part of the process is the unwinding of a double helix. So the double helix is a coiled sort of structure, isn't it? So when it's a bit coiled, it requires some turning to get it to be two straight lines. So that's unwinding for you. Okay, the whole DNA molecule is unwound eventually, but this occurs stage by stage, step by step. Okay, The next thing that happens is helicase. So that's very important to remember the enzymes that are involved here. Helicase, what does it do? It breaks the hydrogen bonds. Now, it makes sense because helicase, double helix, it breaks the hydrogen bonds. It separates the two DNA strands from each other like so. And these two strands will be used as templates to form the new DNA molecule. Now, this process here, in a very casual language, we can say is unzipping of the DNA, but this term is, however, not allowed in the exam. Cannot use unzipping, but you can really say helicase breaks those hydrogen bonds and the two DNA strands are separated. So after they're separated, what we gotta do? We have to build our new strands. So first of all, we need some bricks, right? We need some monomers in order to make a polymer. So here we are having monomers, free activated DNA nucleotides in the nucleus. Now they are free, means they are free in the nucleus for synthesis of new strands. Okay, they're in the nucleus already. There are many of them, ATCG, all different types. And they are made out of the same components as a classic nucleotide, except that they have two additional phosphates. So they have three components: the deoxyribose sugar, the nitrogenous base, and three phosphate groups instead of one that you saw earlier, last video. Now, these two additional phosphates is what makes it activated. Okay, and these two phosphate groups get removed later on, so don't worry about it too much. Now, these free activated DNA nucleotides, what they do is they are actually binding, forming hydrogen bonds with bases on each exposed DNA strands. So again, the two strands act as a template, and these free activated nucleotides form hydrogen bonds with the complementary base pair, which means A binds with T and C binds with G. It's important to note here that no enzymes are required here. It is a spontaneous process. It doesn't require a reaction. It doesn't require energy in the form of ATP here, okay? It just happens. But these are still monomers. They are free activated DNA nucleotides. They are in no way joined together yet. So you can see this G and T, they are about to be side by side, but they are not joined together. They are still separate nucleotides. So we need DNA polymerase to catalyze the formation of a new strand. What it does is that one strand one, right, sometimes more, but attached to each of the two separated parental strands, okay, and this DNA polymerase actually links the activated nucleotides together. And how does it do so? It basically removes two phosphate groups, and you can see the illustration here. And these two phosphate groups can be recycled, no problem, join to another nucleotide and activate it. Okay, and this DNA polymerase also catalyzes the phosphodiester bond formation between adjacent nucleotides. So here we can see the result of this reaction right here in this picture, right? C is bound to T. This here between C and T is a phosphodiester bond. And then this continues step by step as another nucleotide here, you can see this free activated nucleotide is going to complement your base pair, and then DNA polymerase is going to join G to T. Now this occurs step by step, also in the sense of different regions of DNA. So it's not the whole DNA gets unwind and the whole DNA does it at the same time. It is actually section by section. So this section gets unwind and forms first, new strands form first, and then the next section gets unwind. It's a step-by-step process, but this process continues along the whole DNA molecule. Now, I missed out a line here. DNA polymerase also proofreads the DNA. So other than catalyzing the formation of a new DNA strand, DNA polymerase also checks if there's any errors introduced and corrects it before it's too late. Okay, so I think that should be okay so far, right? We have helicase that separates the two strands, these activated nucleotides, the complementary base pair with the exposed template strands. We also call it parental strand, or we sometimes call it the original DNA strand, right? Either way, activated nucleotides do complementary base pairs with them, and then DNA polymerase links the activated nucleotides with the other activated nucleotide, right? And this catalyzes the formation of a new strand of DNA. But to be honest, that's a very basic, simplistic view. And we haven't even talked about DNA ligase just yet. There is a slight complication, though. DNA polymerase is attached to two separate parental strands, as we said just now, but they move in opposite directions, meaning it does not move this way. We imagine that it binds here and it binds here and it moves this way and this one moves this way, right? But the reality is more complicated, like usual, right? It moves in opposite directions, which means one moves left and one moves right. Let's see what that means. Now, there's also complications in the sense of the new DNA molecules. They are always formed in one direction, but as we know, the DNA strands are anti-parallel. So how does this work out? Let's look at it in a visual format. Now, DNA polymerase will move from 3' to 5', always. From this side to this side. Let's look strand by strand, right? Let's look at the strand first. It moves from 3' here to 5'. This entire strand is 5'. So this is 3' to 5'. As it moves, the new DNA strand is formed. And this is anti-parallel, right? This is the 5', because this is 3', so this must be a 5' end, and it's formed towards the 3' end. Now this one has no problem because the process is able to continue as DNA unwinds. As I said, this DNA replication happens region by region, stage by stage, step by step. So as the DNA continues unwinding, DNA will continue moving, DNA polymerase will continue moving, and a new strand will be formed. But let's look at the other strand. Now, the other strand is quite different. The DNA polymerase would bind here or somewhere in the middle. Why? Because again, it moves from the 3', that's 3', towards the 5' end. And in that process, it also forms a new DNA. Like I said, new DNA always form in the same direction, 5' to 3', 5' to 3'. Always remember that. Okay, so new DNA is formed 5' to 3' direction. But you can see this DNA polymerase here, it cannot move any further than 5'. It's at the end already. So as the DNA unwinds, another DNA polymerase is needed to start the process again. So it will bind here, will continue, and as it unwinds, another DNA polymerase will be needed. So we say that one DNA strand is synthesized continuously, and this strand up here is just a nicer picture, is called the leading strand, and the other is called is synthesized in sections or synthesized discontinuously as Okazaki fragments. And you end up with these fragments here because each of these fragments is formed by one individual DNA polymerase, right? This fragment is one DNA polymerase that forms the new DNA strand from 5' to 3'. The new DNA strand is formed by another DNA polymerase here that moves this way. Another DNA polymerase binds here and moves this way, and therefore this is discontinuous synthesis, and this is called the lagging strand, which makes sense because it's like move, stop, it lags a little bit. Like I hope this video is not doing. Right, so it's lagging strand, and that continuously synthesized strand is called a leading strand. And this is when DNA ligase comes in a picture, because we can't leave these Okazaki fragments like this. We need DNA ligase to come in and mend these strands and make it one long new strand. And the result of DNA is, after all of that, is 2 DNA molecules. 2 nice, wonderful double helical DNA molecules, each containing 1 original strand and 1 newly synthesized strand. 1 original strand here, 1 newly synthesized strand in purple here. And that's why it's called semi-conservative. In 1 molecule, half of the molecule, or 1 strand of the molecule, is the original, means semi-conservative. 1 strand is conserved out of 2. Now let me show you how it looks like in the form of an animation to get a closer idea about how it works. Okay, so this is a summary, this is a repeated thing. Okay, so just a reminder, first off, we have helicase that attaches to double helix, and it slowly, this moving slowly, breaks the hydrogen bonds between those 2 strands, and those 2 strands separate. Now, you can see the labels. One is called the leading strand, and one is called the lagging strand. Okay, and you can see that this 3' end is actually talking about new DNA. Right, this new DNA strand is actually being formed from the 5' end to the 3' end. This DNA polymerase is linking the activated free nucleotides here coming in. This DNA polymerase, however, right, is moving in the opposite direction, and is also forming the new DNA strands from 5' end, okay, to the 3' end. And you can see how it moves. It cannot move all together. It has to detach, and then it has to bind again. You can see this DNA ligase sealing the nicks, or joining the fragments between these fragments. What am I saying? Catalyzing the phosphodiester bonds between these fragments to join them up together. And we are almost done here. Here, this one is synthesized. This is a leading strand being synthesized continuously. And this is the lagging strand being synthesized discontinuously. And therefore, I guess it lags behind, I guess. So yeah, you get the idea. The result is 2 DNA molecules, again, each made of 1 original strand and 1 newly synthesized strand. And this is why it's called semi-conservative DNA replication. Now you may be wondering, is there such thing called fully conservative DNA replication, or very liberal DNA replication? Well, it's not called that, but there is something called conservative and there's something called dispersive. These are models that people came up with before we confirmed a semi-conservative. So a conservative model says that, hey, after the first replication, the DNA molecule either has 2 old strands or 2 new strands. Okay, it's fully old or fully new. This is fully conservative or just conservative. Dispersive is another model that we know is not true now, but it was another model that is saying that after the first replication, each DNA strand has a mixture of old and new strands in this manner. And the second replication will look the same. So you get identical DNA molecules and a mixture of old and new strands each. Now we know at the moment that semi-conservative DNA replication is the true one. But how did they find out in the first place? How did they know that it's semi-conservative and not conservative and not dispersive? Well, they did the most beautiful experiment in biology. Well, at least it's called that. And we will talk about that next video. Thank you for listening. Bye-bye.